Hello, good morning. This is Kaushika Patel from LJ Institute of Pharmacy. We have decided to start with the online sessions of odd semesters for the new academic term of 2020. With respect to that, we will start with a subject named Pharmaceutical Microbiology of BPharm Semester 5. Now, microbiology, as the name suggests, is a study of microorganisms. Microorganisms are of micron size. They are very small in size, so they cannot be seen by naked eye. We have to use special techniques to observe them and special methods to cultivate them. There are many different aspects of microorganisms which are not known to us. So we have to study those aspects of microorganisms in this subject. This is very interesting and practical subject, and I hope you all will enjoy it a lot. Now, along with the theory, you also have practicals of this subject. So whatever you are studying in the lecture, you will be practically able to observe them in the laboratory. Okay, so let's begin with our journey of microorganisms. Now, microorganisms, they are of different types like bacteria, viruses, fungi, protozoa, and it is very important to understand the difference between these organisms. So, what we are going to study is we are going to study general microbiology of unit 1. So here you will understand the history, evolution of microorganisms, what are the scopes of microbiology, then what are the differences between prokaryotic and eukaryotic cell, what are the nutritional requirements of bacteria, how to grow them in the lab, how to isolate them, how to preserve them, then how to measure the bacterial growth, and last but not the least, how to observe the microorganisms by using different microscopic methods. So all these topics are the part of general microbiology, and we have to cover all these topics in our session. Now, the first topic which I will like to take is Introduction to Cell. It is very important to know what is the difference between a prokaryotic cell and an eukaryotic cell. Then only you can move further with the identification of a particular microbial cell. Okay? So, what is cell? We all know that cell is the smallest unit that carry out all the processes of life. Now we have divided cells into two types, prokaryotic cells and eukaryotic cells. Now the major difference between these two types is the presence of nucleus and membrane bound organelles. When prokaryotic cell was evolved, it was the first type of cell and it did not have any specific membrane bound organelle. Everything was scattered loosely in the cell. But gradually eukaryotic cell evolved, and in that cell they have a true nucleus and all the different membrane bound organelles. So prokaryotic is pre-nucleus and eukaryotic is true nucleus cell. Now if we study the organelle difference between prokaryotic and eukaryotic cell, okay, then starting with first prokaryotic cell. Now what is present in prokaryotic cell? Inside the cytoplasm, the yellow portion is the cytoplasm, as you can see in the diagram. Inside the cytoplasm, all the organelles, they are loosely scattered. You can find a singular chromosome in the center. There are different ribosomal cells, small ribosomes, they are scattered in the cytoplasm, which play an important role in the protein synthesis. There are different inclusion bodies which are the nutrients present. Then this cytoplasm is surrounded by the first layer, that is the plasma membrane, or it is also known as cytoplasmic membrane. The plasma membrane will be surrounded by second layer, that is cell wall. Now cell wall is a very important structure of prokaryotic cell, and in most of the prokaryotic cells, cell wall will be present. This cell wall will be surrounded by a third layer in some type of bacterial cells, which is known as capsule. Capsule is a transparent gelatinous layer which have some important functions in the pathogenic bacteria. Now surrounding the cell envelope, there will be different appendages that are present. First one is the flagellum. Flagellum is a long hair-like appendage that helps in the movement or motility of the cell. Then there will be short hair-like appendages, as you can see, surrounding the entire cell surface. There will be many number of them, few to hundreds. They are known as fimbriae. Fimbriae is an important organelle that helps in the attachment of a particular cell to a living or non-living surface. Then there are pili. Now pili, they are like fimbriae, but they are somewhat long in the length, and pili can be only one or two in number. Pili, they help in the transfer of the genetic material from one cell to another cell during the process of conjugation. Okay, so now this is a three-dimensional diagram through which you can understand the organelles easily. But if you are asked in the exam to draw a prokaryotic or bacterial cell, then you can draw a simple two-dimensional diagram like this with a proper labeling. It is very important to label properly the diagram, and the arrow should be present in that particular organelle only. You may use different colored pens to differentiate the organelles and the layers. So now if you see a bacterial cell, a prokaryotic cell, then three different types of regions or organelles or structures are present. Starting from the external part, you can see that first thing that is present are different appendages, which are the attachments to the cell surface, that is cell wall. So you can have appendages like flagella, pili and fimbriae. Then second thing is the cell envelope, surrounding of the cell. That consists of capsule, cell wall and plasma membrane. And the third thing that is present is cytoplasmic region. And within that cytoplasmic region, there will be chromosome, then there will be ribosomes, then there will be vacuoles, inclusion bodies, etc. will be present. So this is the structure of a simple prokaryotic cell. Now if you compare it, or if you observe eukaryotic cell, then the major difference you will observe is no appendages are present in eukaryotic cell. Even cell wall is also present in few eukaryotic cells only. Most of the eukaryotic cells have only plasma membrane surrounding the cytoplasm. But what is more important, different membrane-bound organelles are present in the eukaryotic cell, which are altogether absent in the prokaryotic cell. So here you can see that in the center there is a well-defined nucleus with the nuclear membrane, nucleolus and the chromosomes present. Then there are different, you can see here, there are tubular structure which is starting from the nuclear membrane of the cell. This tubular structure is known as endoplasmic reticulum. Endoplasmic reticulum play an important role in the protein and lipid synthesis of the cell. You can find ribosomes attached on the surface of the endoplasmic reticulum. When ribosomes are present on endoplasmic reticulum, it is known as rough endoplasmic reticulum. And when ribosome is absent, it is known as smooth endoplasmic reticulum. Then there is Golgi apparatus. It helps in the transportation process of the cell, and mainly it helps in transportation of proteins and lipids and also in the secretion of proteins. Then you can see this green one, that is mitochondria. Mitochondria is known as the powerhouse of the cell because it generates the energy which is required for the biochemical processes of the cell. Then you can see there is a lysosome. Lysosome is an organelle that contains a digestive enzyme, lysozyme. This digestive enzyme helps in removing all the waste materials from the body. Whether it is excess or worn out organelles or whether it is engulfed bacteria or viruses, everything will be removed from the cell by using this lysosome. Then there will be ribosomes. Of course, ribosomes, they are present either on the surface of endoplasmic reticulum or they may be scattered somewhat freely in the cytoplasm. Ribosomes help in the process of protein synthesis. Some eukaryotic cells, especially plant cells, also have chloroplasts that play an important role in the photosynthesis process. You can even observe centriole. Centriole contains bundle of microtubules and this centriole helps in the cell division of the cell. Then there will be microtubules also in the eukaryotic cell, as you can see in this simple diagram, and this microtubules help the cell to retain its shape in the transportation process, and it also helps in the mitosis process of the cell. So you can see that eukaryotic cells have many different membrane bound organelles present in the cytoplasm of the cell. So if you are asked now, the most important question, differentiate prokaryotic and eukaryotic cell. Okay, so the first thing you can do is you can draw a well labeled diagram of both the prokaryotic and eukaryotic cell in the beginning. Then you can start writing the difference in the characteristics of both the cells in a tabular form as I have shown here. So first, nucleus. What is the difference? In case of prokaryotic cell, nuclear membrane and nucleolus are absent, whereas they are present in eukaryotic cell. Then DNA material consists of only a single circular chromosome in prokaryotic cell without any protein, whereas in eukaryotic cell, different chromosomes are present with proteins known as histones. Then different membrane-bound organelles present in the cytoplasm like endoplasmic reticulum, Golgi apparatus, mitochondria, lysosome, chloroplast, they are absent in prokaryotic cell, but they are present in eukaryotic cell. Then chlorophyll. If chlorophyll is present in prokaryotic cell, it will be dissolved in the cytoplasm, so it is not visible. But if it is present in eukaryotic cell, then it will be present within a membrane-bound chloroplast. Then ribosomes. Ribosomes present in prokaryotic cell are of small size 70S and they are scattered loosely in the cytoplasm of the cell. But in case of eukaryotic cell, ribosomes are large of 80S size and they are mostly bound to the surface of endoplasmic reticulum. Cell wall is generally present in prokaryotic cell and it is of complex composition containing polysaccharides and lipopolysaccharides and peptidoglycan. Whereas in eukaryotic cell wall is mostly absent and whenever it is present, it is simple in composition. Reproduction in prokaryotic cell occurs by binary fission, whereas in eukaryotic cell it occurs by mitotic division. Examples of both the cells you have to write at the end. So most common example of prokaryotic cell is bacteria. Apart from that, rickettsia and few of archaea are also prokaryotic in nature. Whereas fungi, protozoa, algae, animal, plants, all are eukaryotic cells in nature. Okay. So I hope you have understood what is a prokaryotic cell and what is a eukaryotic cell. So when we are studying a bacterial cell, you have to keep the image of prokaryotic cell in the mind. So you have appendages, you have a cell envelope, that is cell wall plasma membrane, and then you have chromosome and ribosomes in the cytoplasm. So, classification of bacterial cells based on morphology. Now, whenever you are studying microorganisms in the laboratory, you are observing the microorganisms using the microscope. The first thing you have to observe is the morphology of the cells. And in that morphology, you have to include three things without fail. First is, what is the size of the cell? What is the shape of the cell? And what is the arrangement of cells in that particular slide? Okay, so now size. When we are talking about size, all the microorganisms are of micron size, but in that size also there is variation in the size range. So diameter of cell is normally between 0.2 to 2 micron, whereas length is between 2 to 8 micron. So when you are writing the morphology, size you can write it down as small, intermediate or large. You don't have to mention the exact size, but you have to mention whether it is small, it is large or it is intermediate. Okay, then the next thing that you have to observe is the shape of the cell. Now shape, mostly there are two shapes which are most common in the microorganism. First is the round or the spherical shape, and the second is the rod shaped organisms. Whenever there are round shaped organelles, it is known as cocci, and when it is rod shaped cells, it is known as bacilli. Apart from that, there are few other shapes like spiral shape, but this cocci and bacilli, they are the most commonly observed bacteria. So now how they are arranged. Now along with the shape, you also have to mention the arrangement of that particular cell. Okay, so if you have suppose observed that it is cocci. Now when you will observe in the microscope, these organelles, these cells, they are very small in size. So when it is round shaped cell, you are just observing a dot in the microscope. Its size will be just similar to the dot. And when you are observing bacilli, it will be like a small line or under dash that you have done. You will not observe a specific rod shape, but you will observe a dash like structure in the slide. So that will be bacilli. And when you see dot, it is cocci. So suppose you have observed dot. Now you have to study the arrangement of that spheres. So if there is singular spherical cells, then the arrangement is single. Then there can be pair of cocci, that will be known as diplococci. Then there can be a cluster of cocci just as the bunch of grapes, that will be known as staphylococci. Then there can be a chain of cells placed next to each other. Now this chain can be straight or it can be curved also. So that chain will be known as streptococci. Then there can be a group of 4 cells on a single plane. That will be known as tetrad. Okay. And when you have group of 4, 8, 16, 32 cells forming a cube in 2-dimensional region, then it will be known as circina. So these are the different arrangements possible for a cocci. So you have to write this arrangement in the observation. Then if you have bacilli, that if you have a rod shaped cell, then it can be singular. Then it can be cocobacillus. Cocobacillus is a shape between cocci and bacilli. So it is like an oval shape. So it is cocobacilli. Then there can be a pair of bacilli, that is diplobacilli. There can be different rods arranged like this, that is palisade arrangement. Or there can be a chain of rod shaped cells, that is known as streptobacilli. Okay, this is streptococci, this is streptobacilli. So along with the shape, this arrangement also helps you to identify a particular species of bacteria. Okay, so here you can see the examples of each type of arrangement is given. So you also have to mention the example in the answer. Now bacilli, they may contain flagella as an appendage. So that becomes flagellate rods. And they may contain spore. So that becomes spore former bacilli. Other shapes, that is comma shaped cells. Once comma is placed like that, shape is that, it is known as vibrio. Then there will be spiral shaped cells. Now if there is a small rigid spiral shape, that is spirilla. And if you find a flexible, thin, long spiral shaped cells, that is spirochete. Okay, so these are the different types of arrangement and shape of cell. So morphological classification of bacteria, if you are asked, then you have to mention this shape and arrangement of cells. So from these topics, what are the frequently asked questions? First, draw a neat diagram of bacterial cell. You may be asked difference between prokaryotic and eukaryotic cell, or you may be asked morphological classification of bacterial cells based on shape and arrangement. Okay, so this is just the introduction of prokaryotic and eukaryotic cell. In our next session, we will continue with the detailed organelle study of the cell. Okay, so in that detailed study, you have to focus on what is cell wall, what is flagella, and each and every organelle you have to study and describe in detail. So that is the ultrastructure of bacterial cell that you have to study. Okay, so in our next session, we are going to start with that part. Till then, subscribe to our YouTube channel Pharma Ignite for the other informative sessions and interactive sessions on pharmacy. Till then, take care. Thank you and goodbye.